So chapter 17 focuses on the cytoskeleton. This is the system of proteins that provide structure and movement inside your cells. The cytoskeleton provides structure, an internal set of tracks for movement of vesicles and proteins and cargo within inside the cell, but also an ability for the cell to physically move by protruding and helping the cell body move forward. It provides a lot of stability as well. Otherwise, your cells would be sort of an unstable bag of goo.
The cytoskeleton is a dynamic network of filamentous proteins that extends throughout the cytoplasm, forming the struts, cables, and girders that give the cell its shape, internal organization, and mechanical support for movement. There are several types of protein filament, each built from subunits that rapidly come together or break apart, allowing these elaborate structures to be assembled and dismantled wherever they're required by the cell. Microtubules are stiff, hollow tubes that anchor other organelles and serve as tracks that guide the movement of vesicles and other cell components. Actin are thin, flexible filaments that form cross-linked bundles and branching networks. Actin is particularly important for cell movement. They are central to the contractile engine in muscle cells and provide the mechanical force for cell movement at the plasma membrane.
So there are three major types of cytoskeleton elements we're going to talk about in this chapter. We're going to talk about actin, microtubules, and intermediate filaments. And as you can see here, we're looking at the top at some staining in actual cells of actin in red, here microtubules in green, and here intermediate filaments in green. Below are some drawings of cells to give you an idea of where each of these structural elements function inside the cell and how they're dispersed, but we'll look at each of these individually as we go through. We're going to start with intermediate fibers, okay? We're going to start with intermediate filaments. These are incredibly strong and provide structure to the cell to help withstand mechanical stress. These are heavily found in hair and nails, but they're also found in all cell types. These can survive salts and non-ionic detergents, meaning that changes in pH or ion levels are not going to disrupt these. They are usually anchored to the plasma membrane at locations that we call desmosomes. You can see here and examples in the figures where they are anchored to the plasma membrane. Intermediate filaments also make up the nuclear lamina, and there are more than 70 different types of proteins that make up intermediate filaments. So this is a fairly large class of structural protein. Nuclear filaments are coded as individual proteins that form an alpha helical design. Two alpha helices coiled together in a coiled dimer. So this creates a twisted rope-like structure. One of the important things about intermediate filament proteins is that their N and C termini look different. They're structured differently. And that means within the coiled dimer, there is directionality, that the N terminus looks structurally different than the C terminus. Dimers interact to form tetramers via non-covalent bonding. And that non-covalent binding makes it sound like the interactions are pretty weak, but the strength comes from the aggregation of so many weaker non-covalent bonds. So we see something similar in the structure of DNA in that two strands are held together by hydrogen bonds, which are not very strong. But when you think of all the hydrogen bonds involved in holding two strands of DNA together across a whole chromosome, in aggregate, that's an immensely strong connection. So similarly here, we have non-covalent interactions holding these together, but the aggregate of those non-covalent interactions provides a very strong fiber. The staggered antiparallel tetramer of two coiled coils. Here you have a coil here and a coil here, and it's staggered in that the ends don't match up. This forms a tetramer. A collection of tetramers, 8 tetramers, come together in a lateral array to create this bundle. And these bundles connect end on end to make the fiber of an intermediate filament. So this is really just an image of a similar thing from a different book, but I think it shows the unique structure of the head and tail a little better than the image from your book. Particularly, remember that I said that there are 70 different proteins that can contribute to intermediate filaments. Those 70 different proteins primarily differ in their heads and their tails. The alpha helix region here in the middle is pretty consistent. So when that coiled coil forms, 2 N termini are together and 2 C termini are together, making what we call a polar or directional dimer of a coiled coil. And then we have that staggered tetramer here, where each dimer, remember, has a pair of N termini or a pair of C termini. And then in that tetramer, they flip-flop. And so then the protofilament are these tetramers blocked together end on end until we get a large mature filament here. And this structure is what has that massive stability to provide the ability for the cell to withstand mechanical stress. There are 4 types of intermediate filaments we're going to talk about. Keratin filaments are highly representative in epithelial cells. Vimentin and related filaments are in muscles, connective tissues, etc. Neurofilaments in nerve cells. And then all cells have intermediate filaments in the nucleus because that's what makes up the nuclear lamina. Okay? You will find more and different types of intermediate filaments in cells that have more mechanical stress. So neurons that have long axons, obviously it needs a lot of stability. So you have neurons that are a meter long. That's phenomenal. Muscles withstand a lot of physical stress. Skin epithelial cells, anything that really is exposed to the outside world, obviously is going to come in contact with more mechanical stress. A network of intermediate fibers helps dissipate that mechanical stress by distributing forces across the whole cell. So the first type of intermediate filament we're going to talk about are keratin filaments. This is the most diverse family of intermediate filaments. Each type of epithelial cell has its own type of keratin that's specific to that tissue. So we're really going to talk about this fairly generically. But these keratin filaments span the whole width of the cell and are connected between cells via those desmosomes we talked about. So here we're just looking at a slightly zoomed in desmosome, which is a connection between 2 cells. This is one cell, and this is the adjacent cell. This whole structure here would be what we call the desmosome. I'm not going to ask you about these specific proteins that make up the desmosome. This was just the best figure I could find to sort of give you the structure. Here we have these keratin intermediate filaments attached at the desmosome in this cell and attached at the desmosome in this cell. And these protrude across the cell this way, and these protrude across the cell this way. Epidermolysis bullosa simplex is a disease in which there is a keratin disruption. And this is particularly evident in skin cells. The skin is incredibly vulnerable to injury and blister formation. And one of the reasons this happens is because mechanical stress can create breaks in these cells, as you can see here. We've got our basal lamina, where these epithelial cells are connected via desmosomes, but the defective keratin network is unable to provide the structure for these cells to stay attached to their desmosome points. And so what you get is blister formation, shown here in sort of a cartoon and shown here in an actual picture from actual skin cells. The next type of intermediate filament we're going to talk about are neurofilaments. These provide structure to neurons. Remember that I said this is one of the types of cells that has to withstand a lot of mechanical stress simply because axons are so long, right? You've got your cell body and you've got your terminus over here, but you've got an axon that's incredibly long and skinny and therefore needs to be pretty stable. There are other cytoskeletal elements along the axons, but it's the neurofilaments that provide the stability for the axon of these neurons. In the disease ALS, you actually get an accumulation of neurofilaments, and it's thought that this might contribute to the neurodegradation and muscle weakness because too many neurofilaments can be a problem and can lead to problems with trafficking and with passing along electrical signals. The next type we're going to talk about is vimentin. This provides a meshy network starting at the nucleus and moving outward. In this image, vimentin is represented in this sort of teal color. Actin in red and microtubules in green. We haven't talked about those yet. We will later this chapter, but those are the other major cytoskeleton elements we're going to talk about. Excuse me. Vimentin is incredibly important in wound healing, plays a role in a number of systems, as you can see here. The truth is it's exceedingly well studied, but there were a set of vimentin mutant mice created that then didn't show any major phenotypes. And so while the structure of vimentin intermediate filaments was really well structured, they made these mice that didn't express vimentin filaments, and they didn't see any major issues. And so there was sort of a debate in the field as to whether or not these were really important. But then further research showed roles in all of these processes and frankly more processes. And so as you can see, a lot of these are really fundamental to the cell being able to move and stay where it's supposed to be and have its primary functions. And so while the mechanism of vimentin's role in all of these has not been elucidated, it's very clear it plays a fundamental role in cell stability. Finally, we're going to talk about the nuclear lamina, which is made up entirely of intermediate filaments. I'd like to remind you that the nuclear lamina is a network of these intermediate filaments that's just on the nuclear side of the nuclear envelope. So here you're looking at the nuclear envelope with a nuclear pore. This is the cytosolic side. This is the nuclear side. The nuclear lamina is a network of these filaments just lining that internuclear envelope. It creates kind of like a mesh. The other intermediate filaments we've talked about are less mesh-like and more individual fibrils, but here it creates a real mesh network that underlays the entire nuclear envelope. This is also important because it's where chromatin attaches. Your chromatin, remember that chromatin is the DNA protein structure that allows all of your DNA to fit inside your nucleus. If all of the DNA in a single cell were totally relaxed and expanded, it would take up about a meter, which is pretty phenomenal how tightly packed that is when you think about the fact that it fits not just inside a cell, but inside the nucleus of a cell. And so chromatin is the structure of DNA that is highly tightly packed and able to fit all up in a nucleus. That chromatin, represented here in brown and here in purple, is not just shoved in the nucleus like at the end of a vacation when you shove all of your dirty laundry back in a bag. It's actually highly organized inside the nucleus, and that is maintained by connections between the chromatin and the nuclear lamina. This is actually basically broken up and reformed with every mitotic event, and meiotic event, but specifically every mitotic event. And that breakdown and reformation is controlled by phosphorylation. So here we're looking at progeria. Progeria is a disease associated with a defect in the nuclear lamina. Here you're looking at just the nucleus of a cell, so imagine the rest of the cell body here. This is in a wild type cell, and this is in a cell of an individual with progeria. And as you can see, the nuclear structure is very different. And this has ramifications throughout the entire body, but can lead to wrinkled skin, tooth and hair loss, cardiovascular issues, etc. So I also want to take a minute and talk about the proteins that interact with intermediate filaments. We call these linker proteins. Particularly, I want you to be aware of cache domain proteins and sun domain proteins. These interact with each other to form a transmembrane complex that protrudes both into the cytosolic side of the cell and the nuclear side.